problems, okay, please let me know as well. Yeah, so that we can take note at least. Like the registration problem, that one, the one who have access to the system is the GO office, and they need to get in touch with Ahmad in Gombak. Okay, I myself cannot do much, but I can follow through and remind them. And if there is anything happen to you with regards to any problems, if we know, then we can help by producing letters and also softly pushing people to move forward. Okay. Right, so we are going to talk about the innate immunity today, and the pattern recognition receptor. Okay, I don't quite like the way the slide appear on my screen, but we will make do with what we have. Okay, so we talked about innate immunity for many times before. I'm sure this is something that you already know. It's not difficult. You are also very close to innate immunity. You see it every day. Okay. The skin, for example, is something that we touch, we feel, we look at every single day, and it's part of the defense mechanism of the body. Yeah. The word innate give the impression that it is not active. Yeah. Something which is passive, which does not change much. Okay, it also gives sometimes the impression that it is not important. Yeah, you look at the skin, you don't see it doing much, right? And everyone is talking about vaccine and antibodies. Yeah, so those things excite people more than something like innate immunity. Yeah, the mere mention of the word innate give this passive, unimportant impression to us. But actually, the innate immunity is as important as the adaptive immunity. Imagine if you do not have skin, somebody who got burned, mangsa kebakaran. If they have more than 40% of the skin burned, they run the possibility of getting very severe infection and loss of the ability to keep the temperature maintained at the correct level. So they become very hypothermic, very cold, and they lose a lot of fluid as well, and they die fast, if they don't have the protection by the skin. So the innate immunity should not be taken for granted. It is as important as the adaptive immunity. A few features of the innate immunity is that the stimulation, activation of the innate immunity does not require prolonged period of induction. Does not require prolonged period of induction. Most of the time, it's already there. It's already ready for any kind of invasion. Okay, if we take the vaccine, for example, which is very closely linked to the adaptive immunity. Some vaccines, like COVID-19 vaccine, they need to be given in a few stages. You need the first injection, and then you need a booster injection. Okay, so between the two injections, you need to wait for several weeks to induce the production of antibody, firstly, and then to enable the memory cells to be produced. And the second booster is to stimulate the memory cell further. So there is a threshold that is passed. Okay, but you don't need this prolonged-ish period with regards to the innate immunity. It also does not rely on clonal expansion of the antigen-specific lymphocytes. Okay, unlike the adaptive immunity. As I mentioned, there is a need for memory cells to be produced. There is a need for the plasma cells to be produced. And once they are activated, they need to replicate in a sufficient amount of numbers, before they can do their work. But the skin, for example, is already there. Once you have an invasion, the leukocytes, you don't need the clonal expansion and the adaptations that take place, so they can work immediately. So whenever we have an infection, the infection itself can be divided into three phases. And the first two phases is where the innate immunity have a very crucial and intimate role. Okay, the first phase is the immediate phase. Immediate phase occurs, sorry, tak boleh, kenapa Nur Akilah?
Tertekan.
Sorry, tertekan. Kalau tertekan, tarik nafas. Kurangkan tekanan. Okay. Okay. Anyway, where did we stop? Okay, I was talking about the innate immunity having a role in the immediate reaction that takes place whenever there is an initial infection or initial wound that occur to the body. Okay, so here we take infection as an example. So when we have an infection, it will be recognized immediately by a preformed non-specific effectors. I'm sure you can name some of these cells already, like the neutrophils, the dendritic cells, so they can immediately attack this infection and then remove the infectious agent. So actually we experience this immediate reaction, immediate extinguishment of the invading agents every day. I will stop from saying that it happens at all time, but it happens every day. Even when we are sleeping, as I'm talking, so this any kind of infection that try to invade our body, within the first 4 hours, we have this occurring without even realizing that the immune system, especially the innate immune system, is working to remove the infectious agent. If for some reason the infection carry on, like when we have flu, sesama, contohnya batuk, some of us have this for some days, so it is more than 4 hours, more than 4 hours it maintains, and then maybe up to 3 to 4 days before the infection is resolved. Some of us may have some fever indicating infections. So in this stage, there are recognition of the Microbial-Associated Molecular Patterns, MAMP, or the Pathogen-Associated Molecular Patterns, the PAMP. Okay, the difference between Microbial-Associated Molecular Patterns and Pathogen-Associated Molecular Patterns is that the Pathogen-Associated Molecular Pattern is more specific than the MAMP. So you can imagine that microbes can be divided into two categories. One is microbes that can cause infection, so we call them pathogen, hence PAMP, pathogenic associated molecular patterns. And then the MAMP, microbial associated molecular patterns. Okay, so if you look at the slide, this I take from immunobiology. The one that can cause infection is the PAMP. So if we are going to criticize the picture, it is at this use of the Microbial-Associated molecular patterns in the figure. Okay, but we understand what is happening. So whether it is MAMP or PAMP, we are not too pedantic about it. So we can still accept MAMP. So this MAMP is the one which acts like the antibody or antigenic receptor for the adaptive immunity. They are the one that then cause inflammation and recruits all the effector cells of the innate system. The final results of the phase is again the removal of the infectious agent. Okay, but here the most of the cells that are involved are of the innate immunity types. Okay. The third phase is the adaptive immune response. Normally, the induction of the adaptive immune response takes more than 96 hours. Yeah. So the infection that takes place involve transport of the antigen to the lymphoid organs. As we have heard in the last lecture, there are two major cells, the dendritic cells and the macrophage, that have the role as antigen presenting cells. So they are the one that present the antigen to the naive B and T cells, allowing them to have or initiate clonal expansion and differentiation from naive cells into effector cells. And finally, the total sum of the whole process is again the removal of the infectious agent. At this stage, I would like to draw your attention to inflammatory response that takes place in all three phases, either the innate immunity, early induced response, or the adaptive immunity. All three will cause inflammation to occur. Okay, so inflammation, what is meant by inflammation is that normally they present with a few features. Can I zoom the slide? Macam tu ke? Is that better? Tak ada respon. Okey ke tak ni? Sorry, saya tak nampak.
Okey.
Sekejap Okay, yes, right. Bila zoom in my window, I can see maybe about 80% of my slide, 70 to 80%. So maybe I need to kecilkan sikit. Okay, right. Where did I stop again? So about inflammation. So all three processes, all three phases, they involve inflammation. Yeah, this is a very important process. The innate the immune system, they need to induce inflammatory response in the beginning. So the inflammatory response have several features. Firstly, normally there is a rise in temperature, secondly, there is accumulation of fluids. And thirdly, perhaps there is redness. On the skin, it's easy to see that there are redness. In the nasal area, for example, if you have flu, etc., and somebody put in endoscope inside, they can see it's very red. In the throat, you can see on the tonsils, it's red. So these are inflammation. Apa tadi? Rise in temperature, redness, and then accumulation of fluid and enlargement of the area. Dia jadi bengkak. So when you are bitten by an insect, mosquito for example, This is a very easy example of inflammatory response that takes place immediately. So you can see it's rise, is itchy, is red, yeah, and it is fluidy, yeah. It is not hard. You can press it, um, but because it's so small, you don't get fever, yeah. But if you are bitten by many bees, for example, yeah, you can have a sharp increase in temperature. Okay. So why is the inflammation very important? Normally, when we talk about inflammation, it's something that we want to avoid. But when we are talking about the immune system, inflammation has to take place. Yeah, it's good to have inflammation in the beginning, because when we have inflammation, what happens is there is an increase in the flow of the blood stream to the area that is affected. Okay? So we have an increase of the blood flow, which means an increase in the supply of the immune cells, whether it is the innate cells or the adaptive cells. Yeah? So all of these cells will be increased in numbers immediately in the affected area. And when they start to attack the pathogen, of course, neutrophils, for example, it will burst. Some of them will burst and then cause damage not only to the invading pathogen but also to the surrounding tissues. So this will then disrupt some of the epithelial lining of the blood vessel, and this is why we see accumulation of the fluids at the affected area. But then we need to contain. We need to contain the inflammatory response. It does not go haywire. Once the infection are all resolved, removed, so the inflammatory response also need to be dampened. Okay, so this question about how the inflammation is aggravated, yeah, from non-existent to stimulation of the inflammatory response, to some aggravation of the inflammatory response, and then to its control. How is this process take place? You will learn about it later on, yeah, when I think in Dr. Ridwan and also Dr. Ariffin, and some part will be covered by Dr. Hafiz, when he talked about hypersensitivity. So the immunology is also about this, about understanding how the inflammatory response is stimulated and then controlled. Okay. Let's move to the next slide. Kat mana saya ni? Innate. Okay, here. Right. Now, the innate immunity is composed of the fixed defenses, like the epithelial surfaces, and the non-fixed defenses, like the phagocytic cells. Something that differentiates the innate immunity from the adaptive immunity is the presence of the pattern recognition receptors. Okay, these pattern recognition receptors, they are receptors, protein-based receptors that is available on the prokaryotic cells. Yeah, the prokaryotic cells, and they are not available on the eukaryotic cells. Okay, and this PRR is what distinguish the invading pathogen from us. Yeah, basically distinguishing self, our own self, from non-self. So this PRR is very important. It leads to the direct destructions of pathogens, and also this is the part that can be presented as antigens to the lymphocytes. We will talk about the PRR later on. And pathogens infect the body through a variety of routes. Firstly, we have the mucosal surface. which involve the airways. Yeah, airways. So the mode of transmission is through inhaled droplets. This can be represented by the way influenza virus invade us, infect us. Okay, influenza virus is very closely resembled by coronavirus. Yeah? All the coronaviruses, influenza virus, they have similar type of symptoms. The airways is also the place where spores of Bacillus anthracis, Neisseria meningitidis can come in and then attack, and they cause diseases like anthrax and also meningococcal meningitis. Okay, and then the GI tract. GI tract is where we get contaminated water or food exposed to the body, and the pathogen involved could be like Salmonella and also rotavirus. Okay, if any of you followed last Saturday's talk by the Biomedical Association of Pahang, of which I'm one of the members, and all of you can also be a member. You can request for the student membership for the society. So during that presentation, the representative from Pahang State, Jabatan Kesihatan Negeri Pahang, Pahang State Health Authority, he mentioned that the highest number of infectious disease in Pahang is actually keracunan makanan. Okay, keracunan makanan, food poisoning, either through contaminated water or through food. So Salmonella is very common. Rotavirus is also very common. They can cause diarrhea. Certain type of Salmonella can cause typhoid fever. Yeah, Salmonella. If you are pregnant, pregnant ladies are also always being advised to eat half-cooked eggs. Yeah, but the risk of getting Salmonella infection in pregnant ladies through half-cooked egg is increased because half-cooked egg sometimes can contain the Salmonella, and because of the level of immunity in pregnant ladies, they can get Salmonella food poisoning and infection. Yeah? So you have to be very careful when you are pregnant. Okay. So apart from airways and gastrointestinal tract, the other mucosal surface which present as a way for infection to occur is the reproductive tract. These come about mainly through physical contact when they are doing sexual interaction, for example. So skin to skin abrasion can happen, so it causes a wound, yeah, tear. So type of pathogens that are common, Treponema pallidum, which can cause syphilis, as well as HIV, which can cause AIDS. And then we have the external epithelia. Firstly, we have skin. Okay, very common is the trichophyton, which can cause athlete foots. This one is mainly when the skin is very damp, yeah, and then we have infection that come through wounds and abrasion, like Bacillus anthracis again, Clostridium tetani, which can cause tetanus, and Francisella tularensis, which can cause tularemia. This is mainly affecting people who handle animals, like in the farms, and if the animals infected by these pathogens, if their skin have wound, they can also get infected by the tularensis bacteria. In the Saturday's talk, they also mentioned about melodosis. Yeah, melodosis is the soil bacteria, which normally live a few meters inside the soil. It's not a surface soil bacteria, but people who are involved in agriculture, if they do not use proper gloves, etc., they use bare hands, they don't use protective shoes, and when they dig wells, they dig their land, they can be exposed to the Melodosis bacteria, okay, and they can get a very terrible recurring infection. Okay, the last one is the infection that can be caused by insect bites. Of course, we know about mosquitoes like Aedes aegypti that can cause dengue in our country. The mosquito in other country, they can cause yellow fever, and then deer tick bites can cause Lyme disease. Anopheles can cause malaria. Okay, so these are the roots of infections for pathogens. So from the two slides, I hope you can appreciate that the epithelial surface is a very, very important first line of defense. It provides mechanical, chemical, and microbiological barriers to infection. Okay, so this is the intrinsic epithelial barriers to infection. Again, in summary, blue showing the airways, and then green showing the respiratory tract, and then yellow showing the gastrointestinal, as well as the sexual organs, reproductive tract. Okay, so the skin, the mechanical defense from the skin, in the skin, is provided by the epithelial cells, which are joined by tight junctions, and also longitudinal flow of air or fluid. And then on the skin, there are also fatty acids. That's why our skin is always moist and oily, because we keep on producing fatty acid. And this is important to discourage bacterial adhesion to the skin. And also it has antibacterial peptides. And on the skin, there are also normal flora, which then compete against the pathogenic bacteria. The same thing can be said of the gut, where they have epithelial cells joined by tight junctions. They have flow of air or fluid one way, from top to bottom. And then the gut have low pH. Most of the part of the gut is acidic, and then it has enzymes, mainly pepsin, which can digest most of the bacteria. Antibacterial peptides also been produced in the guts, and then the gut is rich, very rich in normal flora. The lungs, on the other hand, it has epithelial cells joined by tight junctions, and then protected also by movement of mucus by cilia. There are certain types of disease, like cystic fibrosis, where the cilia produce is there is a disorder affecting the cilia, so the cilia cannot move and cannot when it cannot move, it cannot take away the mucus. So the mucus basically lodge in the respiratory tract and then become thick and restrict the movement of the lungs. So we have to put in some tubes to suck all the mucus. The same thing happens with the COVID-19. The problem with COVID-19 is when they get into the third and fourth phase, the mucus produced by the inflammation that occur in the lungs is too much. So the lung cannot expand and then cannot relax. And this is why they need to have ventilator being put on to help them move their lung. Okay, in the eyes and the nose, again, we can see the epithelial cells with the tight junction is also important there. And then we have salivary enzyme, especially the lysozyme in the eyes and also the nose. Okay, any questions so far? Any question?
No.
Okay. Um, I would like to take 5 minutes break here. Okay, you can go to the toilet or get something to drink. I need to have something to drink, so we stop for 5 minutes and we come back. Okay. Okay, let's resume our class. Okay. So whenever a pathogen wants to invade the body, it has to be able to surpass a few um, a few barriers first, yeah, in the that involve the innate immunity. First and foremost, the bacteria need to have the ability to adhere to a surface, the epithelial surface. Okay, if they could not adhere, they won't be able to invade and break down the epithelial surface and carry on with the infection. So this is why I mentioned in the past slide, yeah, the skin produce fatty acids as well as peptides. These two materials are very useful to discourage adherence of the bacteria to the epithelium. Uh, so uh, the 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 bacteria or the pathogens that are able to adhere to the epithelium, they are able to secrete. Most of it is in the form of enzyme that can break down the epithelium and then causing a wound. Yeah, causing a wound. Uh, Another way is if the epithelial surface is already is already torn or have wound. Yeah, and this will enable the bacteria or the pathogen to get inside the skin or the epithelial surface rather easily. So we have lots of um, ways to protect against this invasion through this manner. Um the wound healing or the wound itself will induce a lot of antimicrobial proteins and peptides, phagocytes and complements that can destroy like microorganism by inducing inflammatory response. And the pathogens that are able to penetrate the epithelium, they will first must be able to initiate a local infection of the tissues. And here they have to encounter and overcome the defense system made up of complement, cytokine, chemokines, phagocyte, NK cells, etc. Okay, so a host of defense system is there. Uh, and if they prevails, and then they need to then face the defense system made up by the adaptive immunity. Yeah, so which are more specific and can adapt to the uh, 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 evolution that occurs or being presented by the invading pathogen. So this is why to invade human is not easy for a bacteria or pathogen. There are a lot of things that they need to surpass, and when we have a chronic infection, okay, it is difficult to treat the chronic infection because chronicity means that they are already able to overcome many of our either the innate or the adaptive immunity. Okay, now how do we recognize the phagocytes? The phagocytes are recognized by some of the innate immunity cells like the mononuclear phagocytes. These mature from the monocyte and they are produced in large number. They present in large number in connective tissue, submucosal layer of the GI tract, and they also present along the blood vessel in liver, especially in the particular cell called Kupffer cells, and also in spleen. Okay. And then the phagocytes are also recognized by the leukocyte, especially the neutrophils, the polymorphonuclear neutrophilic leukocytes. These neutrophils are very abundant in blood, but they do not normally present in normal tissues. But once there is a local invasion of the pathogens, inflammation that takes place will bring a lot of neutrophils to the affected area. So they reinforce the action of macrophages in the event of infection. Now, I mentioned about the PRR, the pathogen receptor, pathogen specific receptor, which is used to discriminate between host and pathogens. So the neutrophils and dendritic cells and macrophages, they are able to attack the pathogen not through an antigen receptor, but through this PRR. Yeah? There are a few types like the mannose receptor, which is mainly used by the macrophage, and then certain ligand like the lipoteichoic acid, which is used by the neutrophils, and then things like CD14 receptor for bacterial lipopolysaccharide, and these are recognized by macrophage as well as the neutrophils. So ligation to the receptors is the one that causes destruction by the phagocytes. The bound pathogen is first surrounded by the phagocyte membrane, and then they would be internalized in membrane-bound vesicle. We call that phagosome because they are going to be phagocytosed by the cells, or they would be internalized in endocytic vacuoles, and some of these may contain lysozyme, so we call them lysosome. Some of the lysosome and the phagosome, they sort of certain vacuoles have a combined function, so we call them phagolysosome. The term phagosome normally means that the vacuole is acidified as well, whereas the lysosome is more specific for vacuoles that contain enzymes, proteins, and peptides. The cells can also produce toxic products like the nitric oxide, the O2 radicals, and the hydrogen peroxide radicals. And these are all toxic to pathogens. So this is the schematic diagram showing what happens. So a macrophage expresses receptors for many bacterial constituents. They have mannose receptors, and then TLR4, scavenger receptor, glucan receptor, etc. So any invading pathogen like bacteria, they will bind to the macrophage receptors. Okay, and then once the binding occur, this will initiate the release of cytokines and small lipid mediators of inflammations. Macrophage will then engulf the bacteria. Yeah, they can form phagosome, for example, and then digest the bacteria. Okay. So this is a list of bactericidal agents that can be produced by the phagocytes. Firstly, the phagocyte can produce or can acidify the phagosome, yeah, and this is either bacteriostatic or bactericidal in nature. And then they can also produce toxic agent derived products like the superoxide, hydrogen peroxide, etc. Toxic nitrogen oxide, okay, such as nitric oxide. And then the phagocyte can also produce antimicrobial peptides like defensins and cationic proteins. They can also produce lysozyme, which dissolve cell walls of the gram-positive bacteria, or acid hydrolysis, which further digest the bacteria which has been acted upon by the lysozyme. Certain phagocytes, they can also produce lactoferrin and also vitamin B12 binding protein. So further reducing the ability of the bacteria to survive and also to become pathogenic. Okay, so this is the phagocytes. If you look at the production of superoxide, this is mainly involving the neutrophils. Yeah? This is generated by a multi-component membrane-associated NADPH oxidase, and it is also known as respiratory burst. It is named so because it involves high amount of oxygen, and then it enables the neutrophil to burst and produce a lot of toxic products that are toxic to the pathogens. This involve hydroxyl radical, hypochlorite, and also hypobromite. So this is an example of how the radicals, superoxide radicals, are being produced. So firstly, we have the NADPH oxidase enzyme, okay, being arranged on the surface of the cells. So they are then activated. Once they are activated, they convert oxygen molecules to the superoxide ion. And then a second enzyme, the superoxide dismutase, convert the superoxide to hydrogen peroxide. In the presence of iron, the peroxidase enzyme further convert to hydrogen peroxide. So the amount of the hydrogen peroxide is increased and then being converted into the hypochlorite ions. Hypochlorite ion, which is very effective against most of the common bacteria. So there is the hypochlorite ions as well as the hydroxyl radicals. Okay, and these are being released either in the vacuoles or through the burst activity of the neutrophils, and then can kill most of the common bacteria. So in essence, we are protected by the common bacteria because of the action of the innate immunity. Now, the inflammatory response that I mentioned is initiated by response of macrophages to pathogens, and the inflammation is important in three aspects. Firstly, to deliver additional effector molecules, so more macrophages, more neutrophils to the sites of infection, and then to provide a physical barrier in the form of microvascular coagulation to prevent spread of infection in the bloodstream. So this is not really, this is mainly hematological, because when inflammation happens, the blood vessel will increase in size. So to bring in more neutrophils and macrophages, at the same time more coagulant to the area, to the area. So increase in the coagulant means a lot of blood cells will become trapped. So they sort of create blood clots in the area where there is wound, and this will then provide a physical barrier, yeah, to discourage bacteria to invade. And then there is a promotion of the tissue repair where the wound would be closed, and all these are dependent on the inflammatory response. So this is to show the action of the cytokines and chemokines, which are produced by the macrophages, which cause dilation of the small blood vessels, capillaries, and also the veins, the arteries. So the leukocyte then move to the periphery of the blood vessels as the results of the increased expression of the adhesion molecules. So the leukocyte, the neutrophils, if you remember, it does not reside in the tissues. It always in the bloodstream. And in the normal condition, the neutrophils just pass through the blood vessel in a very fast manner, yeah, very quickly. But when we dilate the blood vessel, there is increase in the volume, and also because there are also coagulation that takes place, the flow of the blood will become slower, and the leukocyte, especially the neutrophils, is able to then roll on the surface of the blood vessels. And when they happen to bind to a blood vessel receptor, that can bind to their adhesin molecules, they will be able to adhere to the blood vessel surface, and then they have the ability to extravasate, yeah, to go through the tight junction into the tissue, and then expose themselves to the invading pathogen. So the leukocyte then extravasate at the site of infection, and they attack the bacteria. At the same time, the blood vessel will be occluded by the coagulant and the cells. So the spread of the infection cannot become systemic. It will be localized. And once all the infection is resolved, then we have mechanism in our hematology blood system to unblock, to make the blood vessel pattern again. So opening up the blood vessels and to wash away all the pathogens and the byproducts of the immune system. And macrophage dendritic cells can also be can also present the antigens from the infectious agents to the adaptive immune system. Okay, any questions so far? Just to recap, we are talking about the innate immune system. So the innate immune system, they have features which are different than the adaptive immune system. Firstly, in the types of cells that are involved. So the innate immune system does not involve the lymphocytes, and they involve either the APC, antigen presenting cells, or the leukocytes, in particular the neutrophils. So firstly, with regards to the type of cells that are involved. Secondly, in the mechanism of recognizing the pathogens. In the adaptive immune system, pathogens are recognized through their antigen receptor interaction. But for the innate immune system, this is brought about by the presence of the PRR, pattern recognition receptors. Okay, and the difference between the innate immune system and the adaptive immune system is in terms of the length of time needed to induce its action the adaptive system need longer time, okay, whereas the innate immune system, they can act on the invading pathogens immediately. Yeah, they are already there, and they do not depends on clonal expansion. They also do not change very much. Yeah, unlike the lymphocyte, for example. Yeah, the lymphocyte, they have the ability to adapt to the evolution that takes place in the genetic constituents or the physical appearance of the invading pathogens. Something that the innate system does not have. The innate system in general, they have general action. General action. They, although they can discriminate between self and non-self, some of their mechanism can be non-specific, like the neutrophil. When it bursts, it will also kill surrounding tissue and cells as well. Whereas the adaptive immune system, they are very specific in nature. In any way, inflammation, which is initiated by the innate immune system, is very important in the resolution of any invasion by pathogens. So the inflammation is very important, and this is initiated by the macrophage. The innate immune system is also important to activate the adaptive immune system. If we do not have the innate immune system, we have no way to alert the adaptive immune system. So these are the main points of my lecture today. I hope you can take note of this. Yeah. The last part of today's session is on the pattern recognition receptor. Okay. Pattern recognition receptor. Right. Do you have any question? You can type or turn on your mic. Ke semua tidur ni?
Tak ada, doktor.
No,
Ada?
doctor, no.
Okay. Semua dah ada group ke? Have you arranged yourself into groups for the assignment?
Dah, doctor.
Dah. Okay. Can you give me the list? Yeah, the group members and their chosen topic. Okay. Farah can send it through WhatsApp. And then you need to think about the technical aspects of how to conduct the webinar. Yeah? Most likely, it will be only me assessing you. Is that okay?
Okay, doctor.
Right. The last part is on the pattern recognition receptor. Yeah, pattern recognition in innate immune system. It involves proteins which are which form the common features on most of the pathogen. Okay, and the macrophage, neutrophils, dendritic cells, they have also present their own receptor proteins which can bind, which can recognize and bind to these specific receptors on the prokaryotic cells. Okay, so the receptors of the innate immune system, they mediate different type or different functions. Firstly, once the bacteria or the virus, any of these prokaryotic cells, when they bind to the receptors, they would either induce phagocytosis or a chemotactic action, or induce effector cells that can contribute to the induced response of innate immunity and molecules that can initiate adaptive response. Okay, this PRR, pattern recognition receptor. Yeah, this is referring to the receptor that is on the innate immune cells, but the corresponding receptors on the pathogens, these are called the Microbial-Associated protein, okay, or pathogen-associated proteins. You will come across MAMP or PAMP. And there are three types of the PRR, yeah, the protein recognition receptors. They could either appear as intracellular PRR, which means that they are inside the cells, or extracellular, means that they are on the surface of cells and also in their secreted form. Okay, secreted form. Okay. So this PRR, they have several characteristics. Firstly, their specificity is inherited in the genome. There is no such genetic information for PRR in the lymphocyte, so that's why they do not present with this. And the receptor is expressed by all cells of a particular type, like macrophages. It triggers immediate response. They can recognize broad classes of pathogens, and they interact with a range of molecular structures of a given type. However, these PRR, they are not encoded in multiple gene segments. They are only in one particular gene segment, and this is why they do not change. They're sort of fixed, and they could not perform the genetic rearrangement like the adaptive immunity. There is also no clonal expansion, no clonal distribution as well. And they are only specific for certain PAMP. They are not able to discriminate very closely related molecular structures. So if the pathogen change a little bit of their receptor, of their protein arrangement, then the innate immunity will not be able to recognize. So this will be the role of the adaptive immunity to recognize any type of change, any type of evolution. Now, so one type of the PRR is the mannose binding lectin, the MBL, and they present as free protein in blood plasma. They initiate the MBL pathway of the complement activation. The pattern recognition and discrimination from self by the MBL, you need to be careful about this. You need to listen carefully. This is due to recognition of a particular orientation of certain sugar residues as well as their spacing. And these are found only in microbes. They are not found in host cells. What I mean is this. So this is the MBL structure. By definition, the MBL has two clusters of carbohydrate recognition domains. Two clusters. Some will have up to 6 clusters. So the minimum is they have 2. They are all carbohydrate recognition domains, and they have a fixed orientation. So the MBL is a you can consider it as a family of proteins with the carbohydrate recognition domains. Okay, so this is how it then recognizes the pathogens. The MBL will bind with high affinity to mannose and fucose residues with correct spacing. So the key thing is the type of sugar that they recognize, in this case the mannose and fucose, and then this mannose and fucose must be presented on the surface of the pathogen with correct spacing. And thankfully the pathogens, most of the time they have this arrangement. On all the perikaryotic cells, this arrangement is very common. So if the manose and fucose are on the surface but not arranged with the spacing, yeah, the specific prokaryotic spacing, so they will not be recognized by the MBL. So this is how the MBL can discriminate between self and non-self. So on the right, these are all self, and on the left, because they are on the prokaryotic cells, they are non-self. Okay. And the second family of the PRR is the macrophage manose receptor, the MNR. Okay. So this is again by the name, you know that this is again referring to the receptor on the macrophage. So the macrophage has expressed many type of receptors: LPS receptors, manose receptor, glucan receptors, scavenger receptor, etc. So the bacteria can bind to the macrophage receptors and initiate the release of cytokines and small lipid mediators of the inflammatory system. So the macrophage then engulf and digest bacteria to which that bind to them. They can form either lysosome, phagosome, or the phagolysosome. The third family is the scavenger receptor. This is recognized by low density lipoproteins, which has been modified by oxidation and acetylation. They widely recognize and uptake molecules having a negative charge as well as modified uh, lipoproteins. So in the atherosclerotic lesion, macrophages, so these are all recognized by the scavenger receptor. There are three types of the scavenger receptor. Type A is prevalent in macrophages and they have trimeric form. Type B has two transmembrane form, two transmembrane region. And type C, they are transmembrane with N-terminus located extracellularly. So they span the membrane of the macrophage. Yeah. So again, type A is basically the intracellular type, and type B is a transmembrane, and then type C is a transmembrane with a significant part located extracellularly. Okay, the fourth family, the fourth very important PRR family is the toll-like receptor. We call it TLR. Yeah? This is a single membrane spanning non-catalytic receptors. Yeah? It does not stimulate anything. And they are very prevalent. They recognize many pathogen associated molecular patterns. They are also able to stimulate many type of actions in our body. So toll-like receptors are like the TLR1, TLR2, TLR6, TLR3, 4, 5, and 9. Actually, the list goes on. Yeah, so many type of TLR, and they can bind to many types of ligand on the pathogen. TLR1, 2, 6, for example, they can bind to peptidoglycan, lipoprotein, lipoarabinomannan, zymosan. Okay, TLR3 can identify RNA, and then TLR4 can bind to the lipopolysaccharide on gram-negative bacteria. There are also pathogens with flagellin, so these can be recognized by the TLR5. For DNA part of the pathogens, some of these can be recognized by the TLR9. So basically the pathogens, they have to have very sophisticated strategies, yeah, because every part of themselves, every part of the pathogens, human system has has apa ni TLR for them. Yeah. The TLR can recognize basically all types of the parts on a pathogen. Okay. So this is LPS in body fluids bound by an acute phase protein, the LBP. Okay, so they are bound. So the LPS and the LBP complex then been transferred to the CD14 receptor on the surface of the phagocytes. Okay, so once it is transferred, okay, the phagocytes now being activated. The CD14 interact with the Tolerant Receptor 4, resulting in the activation of NFKB receptors in the nucleus. So in the nucleus, and then that will then cause some cytoplasmic changes where the proteins are being presented on the membrane, and then this will then bind to other cells and initiate the production of cytokines, for example. And cytokines, these are mainly secreted by macrophages and dendritic cells. They are very, very effective against many types of bacteria, many types of viruses, and there are also a host of cytokines. Yeah, a long list. They are most of them are very inflammatory in nature. IL-1, IL-6, IL-12. So they enhance the response of the innate system. They also enhance the response of the adaptive immune system. So you can read in this table. IL-1, for example, enhances the response of the lymphocytes, whereas IL-8 they become chemoattractant for neutrophils, so they attract more neutrophils to come into the affected area. This is showing the Langerhans cells with the CD14 and then the TLR4, and then they are being initiated to get into the spleen, to the kidneys. Okay, I think, and then the last point of the that I want you to take home is that the innate immune cells, they are very important for the activation of the adaptive immunity, mainly through the activation of the T cells and then to recruit the B cells. If there is no antigen being presented by the APCs, the macrophage and the dendritic cells, then the T cells will remain naive. There won't be any antigenic peptide being produced, and hence there is no response. But the presence of the innate cells only is not enough. Yeah, there need to be other co-stimulatory factors. Those like the cytokines. Yeah, they need to be produced by the innate system, and it's only in the presence of the cytokines then the naive T cells can be properly activated. Yeah? If there is no co-stimulation, again the T cells will remain naive, and then there is no activation and T cells become unresponsive. We need both the APCs presenting the antigen and also all the receptors being present as well, and then bind to the T cells for T cells to become activated. Okay, so take home message is that the innate cells is as important. In fact, without the innate cells, the innate immune system, the adaptive immune system will not be active. Okay, and finally, this is the last slide, showing the different pathways. Referring to the PRR. So we have the classical pathway, the MB-lectin pathway, and the alternative pathway. So all these works towards activating the complement system, which then help the recruitment of inflammatory cells, opsonization of pathogens and killing of pathogens, and you will learn more about the complement system, the cytokine, etc. In lectures, I think by Dr. Ridwan, Dr. To some extent, Dr. Ariffin as well. Okay, I think that's all that I would like to present for the innate immune system. Okay, so we have the cells, the inflammatory response, the PRR, and then here I have introduced to you the cytokines, as well as the complement, and the fact that without the innate immune system, the adaptive immune system could not be activated. Okay. Any question? Are you all still with me? Tak ada? Okay. I hope you will go to the reference textbooks and read the book, and not only depending on my presentation. There are a lot of things I think which you can add on to what have been presented if you read the book. I would also encourage you guys to go to the YouTube or dailymotion.com and search for good videos, presentations, that can enhance your understanding. If you don't have any question, I will end the session with Tasbih Kafara and Suratul As. Don't forget to sign in for the attendance. Okay, thank you very much. Yeah,